We would like to thank the RPH Research Foundation and the Future Health Research and Innovation Fund for supporting our project. So the blood cancer that we are performing our project on is multiple myeloma. This is the second commonest blood cancer affecting plasma cells, which are a kind of white cells in the blood. And as you have heard, 2,500 patients are diagnosed every year in Australia with multiple myeloma, and at any one time, 22,000 patients are suffering from multiple myeloma and living with it in Australia. The median survival is less than 6 years, and patients suffer from bone pain, fractures, kidney failure, anemia, increased infections, as well as tumors called plasmacytomas. The treatments that we have are chemoimmunotherapy, such as listed here. Patients may also have stem cell transplantation if they are fit enough, and they also may get radiotherapy or surgery. And also, if they have clinical trials available, then we would enroll patients into clinical trials. The average patient will only respond about 30 years to the first-line treatment, and then with each relapse, we would give other medications, and with each relapse, the response duration gets shorter and shorter until the patient dies of the disease. So patients who are refractory to 3 classes of treatment, the proteasome inhibitors, the immunomodulatory agents, and the anti-CD38 monoclonal antibody are called triple refractory patients, and they have a median survival of under 1 year. In the last few years, there has been a new cellular therapy called CAR, or chimeric antigen receptor T-cell therapy, where the T-cells or immune cells in the blood are re-engineered, genetically engineered, to be able to recognize cancer cells and kill them through recognizing a protein called BCMA, B-cell maturation antigen, which is on the surface of the plasma cell. This treatment on the first clinical trial in very severe relapse refractory patients has resulted in an overall response rate of 97%, with patients having a complete response, meaning no myeloma or cancer cells detected, in over 80%. And in the last update of this first study, at 27 months, 70% of patients were still alive. And this really is an outstanding result when we think about the fact that most patients at this stage would have died within a year. Now, this treatment, unfortunately, is not yet available in Australia. The Medical Services Advisory Committee has not recommended its reimbursement by the Australian government, mainly because of cost effectiveness. This costs $465,000 US, which is more than 700,000 Australian dollars per patient, and therefore this is not affordable at its current price. The process of CAR T-cell therapy involves collecting white cells from the patients through leukapheresis or apheresis, and then the T-cells, which are a type of immune cells in the white cells, are then activated. Then genetic modification using a lentivirus to put in the chimeric antigen receptor is then done, and then the cells are expanded in the laboratory. The patient receives some chemotherapy, and then the cells are infused into them like a blood transfusion. So currently, we've had a handful of patients in Australia who've managed to get the BCMA-CAR T-cell for relapsed refractory myeloma through clinical trials. And our experience is that it takes over 4 weeks from the time that the cells are first taken out of the patient before the patient gets the CAR T-cell back. And the problem is that the facility that manufactures the CAR T-cells are overseas, mainly in America. The patient has to wait for a manufacturing slot to be available. It has to be transported by plane, and then wait for the manufacturing, and then by the time it gets manufactured, which takes about 2 weeks, then it gets sent back to the patient. So our proposal is to manufacture place of care CAR T-cells in WA, in the Royal Perth Hospital facility, and we would be able to do this at about 10% of the commercial CAR T-cell cost. And therefore, we'll be able to prioritize treating our patients. We will not have this delay, because in this very advanced phase, myeloma patients, delay results in death. So the CTTWA is a facility which has all the licenses and standards and accreditations to manufacture cellular therapy in Perth, and they will be doing this. We will have the cells collected from the patient through leukapheresis at the hematology day unit. This will be transported to the on-site CTTWA facility, and the manufacturing will be done using a CliniMax Prodigy, which is an instrument which is automated, and within around 12 days we will have the cells available for infusion into our patients. We propose a clinical trial enrolling 15 patients who are triple refractory, and we are having a collaborator who will be able to provide the CAR T-cell vector for us, and they have already published their data on their place of care CAR T-cell targeting BCMA for relapsed refractory myeloma with similar results of overall response rate of 95% and overall survival of over 80% in their group of patients. We also will be monitoring remotely the vital signs of our patients in this study, as well as having a control group of patients who are undergoing standard of care therapy for remote monitoring. So we will be using the Massimo technology, which has a wrist and a chest wall sensor, and we will also be collaborating with the Health in a Virtual Environment Hive facility to help us to monitor our patients remotely. So our hypothesis is that the BCMA-CAR T-cells will improve overall response rate, survival, and quality of life of relapsed refractory multiple myeloma patients compared to standard of care. Our primary endpoints are overall response at 3 months and the rates of cytokine release syndrome and immune effector cell-associated neurotoxicity syndrome, which are side effects of CAR T-cells within 30 days. We also have several secondary endpoints as listed here, and I just want to conclude by thanking my co-investigators and collaborators, and also I wish to dedicate our work to our myeloma patients, particularly one of our advocates who really supported this project, who sadly died last month of relapsed myeloma, age only 56. Thank you very much.